In our density lab, we're going to learn a couple different skills. The first thing that we're going to do is we're going to learn how to use a pump pipette. So the pipette is a plastic or a glass tube right here that is graduated. So it has these different marks on it to help you with measurement. And I've already attached the pumps for the pipettes on the top. So the pumps have a little wheel, and you can move that wheel, and this plunger will go in and out. And as it does that, it's going to extract a liquid, and that liquid will fill the pipette to whichever graduation you want it to stop at so that you get a specific amount of milliliters into the pipette. And that'll help you with measurements. So after we get those liquids into the pipettes, we're going to put those liquids in the graduated cylinder. And the graduated cylinder is where all of our liquids from this particular lab will go. As you read a graduated cylinder, one thing that you're going to notice is with a lot of the liquids, it will form this kind of bubble around the top of the liquid. And that's called the meniscus. And so when you look at the meniscus, you're going to read from the bottom of this curve. And so the meniscus forms by surface tension of the molecules kind of pulling on one another, but also pulling on the sides of the graduated cylinder. So that'll help you with the measurement for looking at the different liquids that are put into the graduated cylinder. We're also going to use a couple different pieces of PPE. So PPE is the personal protective equipment. We're going to wear gloves, and we're going to wear our goggles. And our gloves will protect us from getting any of the liquids on our hands, but also will help us from getting any germs on our hands as well. And our goggles are going to help us in case any of these liquids splash so that we're not getting any of those liquids into our eyes. The goggles will be sanitized at the end of class as well before the next group uses them. So for our density lab, this is what your lab sheet looks like. Like our previous labs, we're going to look at what we know, what we observe, and what we wonder. So remember that when you are looking at know, these are things that you know based on the information that's presented in the lab. They're facts. They're things that you know either beforehand from prior knowledge. It could be something you know after the lab is finished as well, at the conclusion after you see the results. Observe, these are observations that you make throughout the lab. So these are using your senses, except for the sense of taste. So most of these observations will be relying on sight or touch, maybe smell. And wonder is for anything that you are curious about or any questions that arise during our lab. This is the data table that we're going to fill out during our lab. And then at the end for our analysis, we'll circle back to our claim, evidence, and reasoning. These are the procedures for our lab. We're going to take the mass of the cup. So it's going to be a small empty plastic cup. We're going to record that mass. So the mass of the cup will go here. The pipette will be used to extract 20 milliliters of the liquids that are provided at each of the stations. As you put the liquid into the cup, you will take the mass of the cup with the liquid in it. So after you get the first liquid, so for example, this clearish liquid, I put clearish because it has slightly yellow tint to it, just a slight yellow tint. So you'll take the mass of the cup with the liquid in it, and then that will be recorded in this column. Then you're going to determine the mass of the liquid, and in order to do that, we're going to take the mass of the cup plus the liquid, subtract the mass of the cup, and then that gives us the remainder, which is the mass of the liquid. And so that's how we determine what the mass of the liquid is. Then after we have gotten our cup with each liquid in it, we'll take each cup to our tables, and that way we can pour all of those liquids into our graduated cylinder in just a few minutes. So then we'll determine the density of the liquid and determine the identity of the liquid as well. So in our data table, we'll be completing all of this information for each of the liquids. So we'll look at the mass of the cup. That should be the same for all of the cups because they are the same type of cup. The mass of the liquid plus the cup right here is going to help you in determining the mass of the liquid. So once again, to find the mass of the liquid, you take the mass of the cup plus the liquid, subtract the mass of the cup, and that gives you the mass of the liquid. After we find out all of our information, we'll find out the density of the liquid And the volume is going to be standard 20 milliliters, and that is going to help us in also knowing how much liquid to extract with the pipette from the cup at each station. So this is the mass of our empty cup. For remote students, this is the information that you will record in your data table. So if you'll go ahead and do that, and you can pause the video as needed and just kind of go back and forth, or you can pull up the data table in a separate window if you'd like. So 1.6 grams is the mass of just the empty cup. The yellow liquid is next. So these are not in order. So you'll need to look on your data table and find the correct one. So the yellow liquid, you can see the yellow liquid is in here. And the mass of the yellow liquid plus the mass of the cup is 30.2 grams. So in order to find the liquid's mass, we'll take the 30.2 grams and subtract the 1.6 grams. And that will give us the mass of just the yellow liquid. Our green liquid is next. So the mass of the green liquid plus the mass of the cup is 22.3 grams. Again, to find the mass of just the liquid, we'll do 22.3 grams minus 1.6 grams. The 1.6 was just the cup, the empty cup. Our red liquid is 21.6 grams. So you can record that in your data table and go ahead and determine the mass of the red liquid. Our clear liquid plus the mass of the cup is 19.8 grams. And our blue liquid is 17.4 grams. So 17.4 is the mass of the blue liquid plus the mass of the cup. So you should have your data table filled out in terms of the mass of the liquids, the mass of the empty cup, and the mass of the liquid plus the mass of the cup. So you should have those three columns filled out, your first three columns. So what we're going to do next is we'll end up pouring the liquids in one at a time. So this is our graduated cylinder. And since we have 20 milliliters of each of the liquids measured out, then when we pour it in here, it'll just go up basically by 20 milliliter increments. And this is what our result is. So it's a little bit difficult to see down here at the bottom. This is our yellow liquid. And then we have our green liquid, the red liquid, the clearish liquid, and then the blue liquid up here at the top. So that is what our result is. On your data sheet, on your lab sheet, on the left-hand side, there is a picture of a graduated cylinder. So you can get some colored pencils, or you can use a digital online drawing program or paint or something like that in order to draw in the colors of your graduated cylinder. So you can pause the video so you can refer to this, and you're just coloring in the graduated cylinder based on the 20 milliliter increments and based on which colors you see in this density column. So for our density lab, we have completed the extraction of the liquids. We have taken the mass of the cup. We have determined the mass of the liquid plus the mass of the cup. The mass of the liquid itself, if you haven't done that, you can go ahead and pause the video so you can determine the mass of the liquid. It's the mass of the cup plus the liquid minus the mass of the cup. And our volume is standard. We did a consistent 20 milliliters of each liquid to put into the graduated cylinder. So the next step is to look at our density, so the density of the liquid. In order to find density, we're going to look at this equation. Density equals mass divided by volume. So mass is the amount of space that something takes up. So we know that matter is anything that has mass and takes up space. So mass is just a measurement, a number that quantifies how much stuff is in the matter. And the volume is a reference to the amount of space that something takes up. And we were able to determine the volume by looking at the measurements in the graduated cylinder and also using our pipette to extract the 20 milliliters. So our mass is going to be measured in grams. So that was the information we were able to gain from the first three columns of data. And our volume is this column, which I already pre-filled for you, the 20 milliliters. So the volume is measured in milliliters, ml. So this is the short form for the equation, just abbreviated. Density equals mass over volume. So then the next part, after you have determined the density of each liquid, the next thing we'll do is determine the identity of each of the liquids that we were provided. So we know the color of the liquids because it's listed on your data table. And then I provided you with a graphic here that has some common liquids and their approximate densities. So after you have done the math and determined the density of each of the liquids from the lab, then you'll look up here at this chart and you will determine the identity of each of these liquids based on the density of the liquids. In Canvas, this is what your data table looks like. So this is all fillable text boxes. So this is, I think I actually put this on 2 slides for you so that you would have a little bit more room. So here is the graduated cylinder that you will fill the colors out on, and remember you can use a paint program or something like that digitally, or if you prefer, if you have a printer at home and you want to print it out, that's fine. I don't, and so I would just fill that out digitally. But all of these yellow boxes, these are all fillable online, so you can just type straight into this. And then here's your data table right here. So make sure you have all your information on here. And as you're filling in your data table, make sure you include your units. Mass of the cup is in grams. Mass of the cup plus the liquid is in grams. Mass of the liquid is in grams. So all of those are mass. They're all standard grams, and that's what our balance that we use to collect that information was using. The density of the liquid is going to be in grams per milliliter, because remember that it is measuring the mass over the volume. So the mass was measured in grams, the volume is measured in milliliters. So that's why it's gram per milliliter. And then the identity, you'll just have the name of the liquid listed here. So then our next section is our claim, evidence, and reasoning. So when we do CER, when we do claim, evidence, and reasoning, this is going to refer straight to the information that we collect from your lab. So our claim is going to be referring to what question or what problem we were examining throughout the lab. So your claim is going to be just a statement that you believe to be true based on the information that you collected, based on your observations, based on your evidence, based on any qualitative or quantitative data that you may have gathered. It's not necessary to say the question is, or the problem is, or the answer is. Just write a sentence that explains a statement. Just one sentence probably will do. So just a sentence that is a statement that is true based on your observations and based on your evidence in the lab. Your evidence, these are your clues. The evidence is going to be straight from your observations, which is why we look at what you know, what you observe, and what you wonder. And this will help you in order to analyze what happened during the lab. So the evidence will be your clues. It's data, and data can be quantitative and it can include numbers, or it can be qualitative and it can be all observational using your senses, except for the sense of taste. So this is research that was gathered, data that was gathered. And sometimes it may be from a video or an article or something like that. But in this case, it was done throughout this lab. So you'll list your evidence here. Bullet points are fine. A sentence is also fine as well. But the evidence is just straight from observation, straight from the lab. It's not an explanation. It does not include because or some reference to the explanation as to why something happened. It's simply just the evidence that led you to this claim. And then your reasoning is why. So your reasoning is the explanation. So it's how the evidence answers the question or solves the problem. There's no need to put this solves the problem or this question provides evidence. Be specific. Refer specifically to pieces of evidence. Refer specifically to the claim. So you don't even need to use the word reasoning. It is okay to put the word because. It is okay to link the evidence to the claim because the evidence should be supporting your claim. So the reasoning is going to be a couple of sentences, probably a paragraph in length, so 4 to 5 sentences that are good, solid scientific sentences that explain how the evidence specifically supports the claim from your lab.